who was initially trained as a biochemist and who is today's speaker. So during his academic career, he worked as a German Max Planck Society and also at Princeton University, where he developed quantum chemical methods that allow for the accurate prediction of molecular properties and which can be used in combination with or even replace experimental data. And he developed a growing interest in his work for the pharmaceutical and chemical industry. So he co-founded the company FACCTs that he's going to talk about today. So it's FACCTs GmbH, using the German terminology, and he's now the managing director of this company. So it promises to be really very interesting. So now with FACCTs, he now leads the further development of new and groundbreaking predictive methods for life science applications, including spectroscopy, catalysis, and collaborations with pharma partners. So many thanks, Chrisoph, for accepting to do this. The floor is yours, and you have, plenty of time. So,
perfect. Thank you very much. First, I want to ask, is the sound okay? And do you see my slides? Does it look?
I can see the slides, yes. Is that, yeah, and we can hear you, I think.
So, thank you. Yeah, thank you for, I think we met last year at QM and Pharma meeting in London, and it's great that, this started an interesting conversation, which led to the talk today, and hopefully some more events in the future. And what we are doing at FACCTs is we do high accuracy quantum chemistry, and that's, as you explained, is capable of predicting experimental results or compatible results comparable to experimental data, and thus enhance experimental data and also try to pre-screen and use that in also industrial R&D. And my talk consists of two parts, and for the first part, I would like to show you this figure here. So that's, I'm trained biochemist, so I'm interested in proteins and biosystems. And what you see here is a protein ribonuclease, which has a bound ligand in the active site, which is quite unusual. It has a lot of negative charges, as you can see here. And in the last years, or last 10 years or so, like we and also others, of course, invested a lot in speeding up quantum chemistry calculations. And so we managed to really make those high accuracy QM calculations affordable, meaning we can nowadays tackle systems like these at a very high level of accuracy. And so in the next 20 minutes approximately, I will try to show you that we can now analyze these binding modes of such a protein ligand interaction with standard calculations nowadays. In the second part of my talk, I will show you some more real-life applications that we got from pharma partners, which showcase how they are using high accuracy QM calculations in their production runs, in their daily R&D. But first about myself. So FACCTs, I'm representing FACCTs here today. FACCTs is short for fast and accurate computational chemistry tools. And our mission is to bring exactly that to industry. There's a lot that happened in the last years in fast and accurate QM development, especially also in the team of Frank Niese, where Orca is developed, and Orca is our main workhorse, and it's a molecular quantum chemistry package, which you can use for those who are not familiar with QM. You can use that to, for example, do simple things like plotting molecular orbitals. You can also compute structures, or predict how does a molecule look like in gas phase, or how does it look like in solvent, in water, or in any other solvents, in DMSO, chloroform, and so on. And you can look at conformates. You can look at what are possible conformational states of my molecule. But that's just one tiny part. You can also look at reaction mechanisms. So quantum mechanics, in contrast to force fields, which probably a lot of people here know, quantum mechanics can break bonds and can form bonds. So we can do reactions between different molecules. We can bind two molecules to each other, and we can analyze how does that reaction take place? How does the structure look like when going from the reactant to the product state? How does the transition state look like, and what are the energies associated with that? Other aspects are, for example, we can look at spectroscopy. So quantum mechanics copes with how do electrons move around nuclei in a molecule. And so we can look at different electronic properties. So exciting electrons in excited state, but also looking at the interaction of an electron with a nucleus, and so on, and how they interact with an external field. And that can be NMR, for example, or that can be ECD, VCD, and UV-Vis, IR spectroscopy, and so on. So we can use quantum mechanics to model things like spectroscopy. And we can do that for very small molecules like here, but we can also do that for large molecules like proteins, active sites in a protein. If we have metalloproteins, how does how does the metal look like within such a protein? And that's a new thing. ORCA is a molecular code, but we can also look at periodic systems. So solids are usually, have some periodicity, and so we can also do that. But I think that's out of interest of the current discussion here, because this is about biosystems. Here I'm listing the available features that ORCA has for those who know about quantum mechanics. For those who don't, just to give you a high-level overview, there's a lot of different methods in quantum chemistry. There's semi-empirics, they're usually very fast, but also less accurate. And there's also things like couple cluster theory, multi-reference methods, which are much slower. Much slower means like orders of magnitude slower. It means like you have a system and you can compute it with a semi-empirical method in seconds or milliseconds, and a multi-reference calculation on that same system can take weeks or months. And ORCA has very efficient methods, especially for these high accuracy methods. And we will have a look at that later. System-wise, we can look at molecules, large biochemical systems, solid-state systems, and just to show you what ORCA is especially good for, that's spectroscopy. So people, especially also in the lab, are using ORCA a lot, because when they want to compare their experimental results with computed results. And reactivity, that's, as we see in this figure here again, how do transition states look like? How is the thermodynamics of a reaction? How is the activation energy, activation free energy, and so on? So that's a rough overview on the capabilities of ORCA. Behind ORCA, there is, I mentioned Frank Niese already, there's a large team within the Max Planck Society. So Frank, he's a director within the Max Planck Society, and he's heading about a large team of 30-40 people, and a lot of those are working on full-time ORCA development. Then the FACCTs team has grown over the last year. So we were founded in 2016, and we grew organically until now to about 20 people, and we are working closely together with our industry partners. So ORCA is free to use for academics, and they can get ORCA from the Max Planck Society or also from the FACCTs homepage for free. But we earn our money with license agreement that we have with industry people. So whoever uses ORCA to earn some kind of money, so exploit quantum chemical calculations commercially, also has to give back, so to pay license fees. And also ORCA development is supported by a large collaboration network around the globe. So we have about a dozen academic collaboration partners for those aspects where the team here, the core team, where it's not in their own interest. We have a very close collaboration also with the Grimme group. So Stefan Grimme, he's, as Frank and the other people that you see here, he's a scientific advisor to us. So he's very well known for his XTB development, dispersion corrections. And we at FACCTs, we are based in Cologne, which is in Western Germany, where most of you know about Cologne. Mülheim is about an hour apart, and Bonn, where Mülheim is where the Max Planck Institute is, with whom we collaborate. And Stefan Grimme, his group is based in Bonn, which is half an hour, right? And so a lot of the people that you see here in the team also are trained quantum chemists which come either from Frank's group or from Stefan Grün's group. But we also have other people, so from other groups, and also non-quantum chemists, physicists, computer scientists. Yeah, all this, yeah, powerhouse in terms of development and expertise in terms of scientific advices has led to a large popularity growth in the last years. And this we measure usually yearly by counting the number of citations to ORCA and also to the competing programs. So a lot of people in academia use ORCA, and they always cite, yeah, write papers and have to also cite the programs that they're using. So they're not just using ORCA, they're also using other tools. And what you see here is the ORCA citation share over the last 10 years. And you see that this citation share is growing quite nicely. And you see also that we are not yet number one. So, but we are on a good way to maybe or probably become the most popular molecular quantum chemistry program. We have now almost 100,000 registered users, and we are working together with more than 60 industry partners. Just to give you an overview here, so we aren't allowed to show a lot of the logos of our clients due to confidentiality agreements, but we can show them for some of them, and here you see like a good representation. So we work together with big pharma, biotech companies, chemical industry, material science companies, and also some technology companies. And we also have quite some users and industry partners in the UK. Now, that was about facts and also rough overview on ORCA. Now let's dive deep into high accuracy quantum chemistry. High accuracy quantum chemistry can be either DFT, density functional theory. That's a method or concept for which also the Nobel Prize was awarded in the 90s. And that's one, like the main pillar of, or the main workhorse for quantum chemists when they run their for this, for high accuracy DFT, we worked a lot, especially that started over the COVID crisis. We worked a lot on making these calculations better within ORCA, so making them faster, more robust, and also more memory efficient. ORCA was already a very decent program until then, but what we did, like, you have to know, quantum chemical programs, usually they develop over years, over decades, and then a lot of PhD students work on them and implement their things. And so like there's code-wise scientific development is interesting. People do a lot of complicated stuff, but they are not trained computer science. And so we want to have a stable, very stable, solid base for our ORCA, because that's also the foundation of this company. And so what we did, we invested about 3 years into rewriting the entire architecture and backend behind ORCA, so that we can unify a lot of those things within ORCA and also on the go speed up things and bring down the memory requirement. And the result of that, we released in 2024 with the ORCA 6 release. And when doing that, we saw that we had quite some significant improvements in speed for our program. And so what we were quite curious, okay, how did ORCA actually, how did the speed develop over the last 20 years? And the very first ORCA version that was released was to broader public was in 2004. That was version 2.4. And we have internally, we have like a Git versioning system where we can take the different release versions, the source code of the different releases. And so what we did was we took those different release versions, the source code, and compiled these versions on a new state-of-the-art computer with new state-of-the-art libraries. And libraries here is quite minimal because ORCA just needs BLAS/LAPACK, which we need for matrix-matrix multiplications, and also OpenMPI, which is what we use to parallelize calculations. And we did this on the same computer in order to really measure how much more effective the algorithms became within ORCA. And we used a high accuracy standard which is hybrid DFT and triple zeta base. So that's a term which is, if you ask a quantum chemist, which is something that is decent and that people would use to predict properties and to predict energies. And here you see a molecule which is about 150, 160 atoms large, and that's something that people like in 2004 would never have computed on that level of theory, because it's just too large. But now we did the experiment on a new computer, so we had a fast computer and we had these different release versions. And the speed up you see here, which is quite significant and astonishing. So the 2004 version has a speed of 1, and we see with Orca 6 we had a speed of 160 with this new version on the same computer. And now, of course, in those 20 years, also the hardware was improved significantly. And I was actually quite astonished when I saw this. Like, if you look at the single thread speed up, that's just a factor of about 10. So of course, over that time, computers became larger and larger. We get like more and more parallelism. So we have, at that time, we had probably 1 or 2 cores, dual cores. And nowadays we have easily computers, CPUs with dozens, 60, 120 cores. But if you look at the single thread, at a single core, then the speed up is just a factor of 10. If you combine both, then you get to a speed up of about a factor of 1,000. So if you compare the calculation that you did 20 years ago on an, at the time, state-of-the-art quantum chemical program and state-of-the-art computer with a nowadays state-of-the-art computer and state-of-the-art QM program, then you get a speed up of a factor of 1,000. That means in 2004, if the calculation took a month, it now just takes 30 minutes. And that really changes the way people are thinking about things, the way what is possible in terms of what do I do in my lab or in my computer lab. So what types of calculations do I run in order to, because these calculations that people in industry, which is our target group here, if an experiment is faster, is cheaper, then they do the experiment. Only if the computation really helps them in terms of cost, then they would also run that computational experiment. And of course, it also needs to be, if it's taking a month, it doesn't help, because nobody will, like, after a month, like, an entire strategy has changed. So really, speed here is of big importance to get things done now, get results now, and also to have a low cost, which is comparable to the experimental data cost. Just to put some flesh to the bones here. So what does that mean for average pharma molecule? So here we have a 60-atom system, and there's a lot of different flavors in DFT. So there's R2Scan3C, which is a method which you can use to run optimizations, and such a calculation on 60 atoms takes about 20 seconds. If you go to a very good functional, which is what people would use nowadays, that takes about 1.5 minutes. And if you go to a post-Hartree-Fock methods, these are, we will talk more about that later, high accuracy wave function methods, then that calculation takes about 12 minutes. Now we can go larger, we can go to 500 atoms, and these calculations are also feasible. So then they take like about an hour. And if you go to even larger molecules like proteins, and that's what people here are interested in, then we can also compute those. And this was not possible a few years ago. I need to emphasize that here. So these calculations, they take just a few hours. So that's possible nowadays. And with these speedups, so speedups in terms of high accuracy calculations on a large scale, on systems of hundreds of atoms, and robustness, really, people who know quantum chemistry, they know that quantum chemistry is known to things fail. Like SCF doesn't converge, something goes wrong, and that's also like a barrier for people to use quantum chemistry. And with our entire rewrite, we really brought the quality management into Orca, brought robustness to Orca, so that really a lot of calculations are out. Almost 99.9% of the calculations run through and give you a meaningful result. And that was also the motivation for, like, in the last years, machine learning had a big rise in computational chemistry. And a lot of people produce, for example, machine learning potentials. And for these, you need data sets. So they use either experimental data or quantum chemical data, because quantum chemical data have the advantage that they are clean, so they are reproducible. And so what people do is they run thousands, hundreds of thousands, or in this case, millions and hundreds of millions of calculations in order to create a data set that then can be used to train a machine learning model. And so what Meta did over the last year was they produced a data set consisting of more than 100 million high accuracy DFT calculations on a large variety of molecules. You can see the target applications down here. And that's quite impressive, with an unprecedented size also for the individual system. So they went up to 350 atoms, covered almost the whole, almost all the elements, and did not stop at organic molecules, but also went to metal complexes, which are usually quite complicated. So if something goes wrong in SDF convergence, then that's happening with metal complexes. Plus charged species and different spin states. So when we have a metal, then especially transition metals, they can exist in different spin states that can be low spin, high spin, and so on. So there's a lot of details that have to be looked at there. And so they did this with the help of Orca. So they ran, I think if you look in the paper, look it up in the paper, they ran about 500,000 computers for over a year. So that's massive data, a massive compute that went into that. And so our contribution here is like this data set, like all these quantum chemical calculations were run with Orca. And these systems were quite large. So as they also acknowledged in their manuscript, was that Orca really enabled these types of calculations for these large systems at the high accuracy level. And also, and I want to emphasize that here, if you run 100 million calculations, you need to have decent robustness, so that like all these calculations, or most of these calculations, really run through so that you get a meaningful result. And so that's just another perspective to this. So if you want to create large data sets, then you need a robust program and a very efficient program, because otherwise you spend, if the calculation takes 10 times the time, then if you don't have 10 times the amount of computers, like 5 million computers, then it would just have taken a much longer time. So years, decades, or even hundreds of years. So we just talked about DFT, high accuracy DFT, and there's a different pillar in quantum chemistry, which is post-Hartree Fock method. So DFT is the main workhorse here, but post-Hartree Fock methods, they're actually known for even longer, but they were, until recently, they were not that usable because of speed limitations. And here I want to mention for single reference cases, CCSD parenthesis T, which is for these cases known as the gold standard, or widely accepted as the gold standard in quantum chemistry. And you can use it to produce conformational energies, reaction energies, activation barriers, and also electronic excitation and spectra, which are really in line with experimental data. And this for organic compounds, but also for a lot of transition metal containing systems. These calculations, they scale with n to the power of 7. That means if you double the system size, the calculation will take roughly 3 orders of magnitude longer. And so that becomes, they become prohibitive at about 20-25 atoms. So what we in the Orca team developed a few years ago was the DLP-no approximation, which brings the scaling down to linear scaling. So you reach system sizes that are actually of interest to academia and also to industry, and at a similar cost instead of DFT. So these calculations are just maybe a maximum an order of magnitude slower than DFT, but they are much more accurate. So they bring the failure rate down by a big factor, as you can see here. And so following that publication or that initial release, since then, there's been a lot of people, especially also experimental colleagues, who have benchmarked these methods and also found that they are really producing results within the experimental error bar. And also just to emphasize that Frank's team, they have worked a lot in the last year since the very first release in order to make, to also use the DLP-No approximation for a lot of other methods so that we can really have, can look at realistic system sizes with the entire method arsenal that is out there. And here you see an overview on the methods that we have now for the DLP-No approximation available. You can look at energies, but also excited states, and also at a lot of properties. And that's something we will talk more about later, at energy decomposition. So that's something that quantum chemists, that can be useful not just for quantum chemists, but also for experimental colleagues, which can use this in order to understand how do different systems interact with each other, and what's the interaction strength. So that's something we will look at next. But first, you remember this protein that we had a look at, ribonuclease A. That's a big protein, has about 2,500 atoms, and we are not there yet that we can run couple cluster calculations on such a protein. But what we can do is we can use Orca's multi-scale models. So multi-scale models, that's also where there was a Nobel Prize about 10 years ago about multi-scale models, PMMM. They make use of the fact that usually people are interested in a small part of a large system. So we have a local phenomenon that is of high interest, and so we can use a high accuracy method for this part of the protein or whatever system we are looking at. And for the big part of the system, we take a lower level method. So that's multi-scale, different scales at different accuracy. And so in Orca, we have different multi-scale schemes available, and we can run, for example, also reaction mechanism calculations using multi-scale schemes, but also we can look at protein ligand interactions. And for example, we can use couple cluster for the high accuracy and for the active site, and we can use lower levels of theory for the surrounding. And now let's look at ribonuclease. So to explain to you what that is, so this is a protein, an enzyme which digests RNA. So that's a problem for a lot of people in the lab who are working with RNA. And so it's of high interest to get an inhibitor to RNA so that it doesn't digest all the RNA. And there is a natural inhibitor, which you can see here, which is not that easily to produce. And so Kit Cummins, who actually did the work that I'm presenting now here, he and his team, they worked on finding inhibitors to RNAs. And so in a first paper that they published in 2019, they found this inhibitor, which is similar to uracil and which has 5 phosphate groups. And it was amongst a series of different uridine nucleotides, P5U for the 5 phosphate groups, like the strongest inhibitor. And so what they wanted to look at in the current work that I'm showing here is they wanted to see whether that trend continues. So when they add another 1 or 2 phosphate groups, yielding P6U, P7U, does that trend continue? And so they did experimental work, crystal structures, inhibition assays, and they also did computation. So they ran these multi-scale calculations using couple clusters theory for the ligand plus close residues, about 300 atoms, and they used a lower level of theory, HF3C for the surrounding. And overall, what they found was that the experimental trend was in line with the computed trend. So the P5U was the strongest inhibitor, and also they could reproduce that with the calculations. But it was just like the start of the story. So the more interesting part is these are ligands that have like very highly negative charges. So it's interesting to understand what the actual binding mechanism is here. And that is where we can make use of the local energy decomposition scheme. So that's something we have in Orca in order to take this highly accurate couple cluster, DLP-No couple cluster energy, and decompose it into the interaction energies between different parts of the system. That's something that can be useful for a lot of different applications, and we will use it here for the protein ligand complex. I want to explain to you what that means on the basis of this simple example. So this is a water cluster with 6 waters, which are interacting in some way. So we see a lot of hydrogen bonds here between the different waters, but also some of the waters are not interacting at all. And here on the right side, we see a heat map of interaction energy. So that's something we get out of these LED couple cluster calculations. And what that means is we can look here at, for example, water 6, water 5. So we have all these waters here and their interaction energies on that heat map. these two waters, they have a short hydrogen bond, which usually means also they have a strong interaction energy. So that's minus 10 kcal per mole, which is strong. Now we can look at the different water pair, water 2, water 5, far apart and no hydrogen bond, and also like a negligible interaction. And now the beauty of this method is that we can even have a closer look into each of these interaction energies. So we can take that water 5, water 6 interaction energy and decompose it further into its components like electrostatics, exchange, dispersion, and charge transfer. And that's something that if we now look at protein ligand interactions also becomes very useful, because there we have a lot of unusual interactions like sigma hole interactions and so on. And it can be quite interesting to analyze those interactions in a protein environment. Now we can build models, and that's what they did for these different ligands with the proteins, and they also had the crystal structures available, so that was quite straightforward. And then also produce these heat maps. And what you see here is 1 to 11. These are the active site residues, and P6U in this case is the ligand with the 6-phosphate group. And we see here the interaction energies between the ligand and these active site residues. And now we can do that for the different ligands for the entire series here. And we can take those first lines, which are ligand interaction with the different residues, and we can look at these interaction energies. And you see some are quite stable and others differ quite a lot. And so now what they also did in that paper is, and here I'm just showing some examples, they looked at these different interaction energies. And for example, you can look at the lysine interaction with the ligand and check how is the trend of interaction energy here. And you can do the same also for histidine. And that way you can try to understand why does P5U, why is it the strongest interacting ligand here? Why is it the strongest inhibitor? Now, what we did over the last 1-2 years is we went a step further in terms of method development. So now we have this ligand here. This is just one of those ligands in the active site. And now what about if we can go and split up the ligand actually into its different fragments, which are functional groups of this ligand, and have a look actually how does this particular functional group interact with this particular residue? And so that gives us information on each individual functional group and what its contribution is to the binding to the protein. And that can help us also to think about and design new ligands. And so that's where we, over the last 2 years, developed the so-called covalent method, which means covalent LED, and I'll explain the concept next. Here we do this, explain it for the simple ethane dimer. And so what we need to do in the covalent framework, we need to split up the system into 2 fragments. And so here we have each ethane split up into its 2 methyl groups. And then what is happening in the steel-pinot couple cluster approach is that we localize these molecular orbitals, assign the electrons in there to the different fragments, and then compute the interaction energy and distribute it to the different fragment pairs based on these assignments. And so in the standard LED approach, if we have here a bond situation, then we also have we have a bonding orbital. And in the standard LED approach, this orbital and its contribution to the energy is assigned to one of the fragments. And now in a covalent approach, it's assigned with some weighting factors to the 2 different fragments. And now we can look at and compare the energies here. And so this is for this ethane dimer. You can see here on the left side the standard LED approach, and on the right side you see the covalent approach. And you can see that here on the left side we have quite unphysical interaction energies. So we can look at 1 and 3 and 1 and 4. You can see here we have interaction energies on the order of 100 kcal per mole, which is not physical. And so now with the more balanced treatment, we get to much more reasonable interaction energies, which can then be used in order to interpret the results. And that's, of course, the ethane dimer is not too interesting, but we can look at biomolecules like the DNA double helix. And that's what we see here. So in the DNA double helix, we have why is there a double strand? How does it interact with each other? We have stacking interactions, and we also have strong hydrogen bonds between the nucleic acids here. And so that's what was done in these first calculations here. So we have a double strand, we have about, yes, 7 of these nucleic acids interacting with each other, and we can split it up in the way as you see here. So we have the different nucleobases here as fragments, but we also have the entire backbone as another strand. And when we can construct this heat map and analyze the interaction energies between those different nucleobase pairs. And you can also directly see that we here on the diagonal we have, for example, 14-1. So these are hydrogen bonding, and you see also the strong contributions from the hydrogen bonds. And guanine cytosine, they have 3 hydrogen bonds, so that interaction is much stronger compared to the adenine thymine interaction, which just has 2 hydrogen bonds. We can have a look at other examples like guanine riboswitches, which is some kind of RNA which has a bound ligand, and here now the importance of the covalent also comes into play, where, or like the potential. So we can take this ligand, so that ligand here is shown in this 2D figure interacting with the different nucleobases, and we can now split up the ligand into different fragments, and then also and now can check, okay, how do these different function groups interact with the surrounding? And the nice thing here is, like, for RNA, there's no established, or the force fields that are available for RNA are not as established and as robust, as good as for example for protein. So really having a robust, accurate methods here that give us good numbers has quite a big potential here. And so now we can take this example and also construct these LED maps, these heat maps for the local, using this local energy decomposition framework. And then I won't go into any detail here. I just want to show you that we can then check, okay, how do these 3 different fragments of the ligand interact with the surrounding here. Now we see that we have to, like, of course, we don't want to manually go and construct all these different fragments. And here I just want to point you to our new Orca Python interface, and which, like, Orca is a text-based tool, so you need to write input files. And over in like last spring, we released an open source tool that's also available on GitHub, and where we also encourage people to contribute. And this tool allows you to run Orca calculations from a Python environment. And we have also already published quite a few examples in the form of Jupyter notebooks, so that you can run a lot of different standard calculations from the Python level. And that way also use the entire environment, a lot of other libraries that are available within Python. And within other Python packages. And this also, just as a hint, this also can be used quite nicely for teaching. And it's a lot of people are starting to use that. And what we did for these covalent calculations is we created Jupyter notebooks which automatically create reasonable fragments for different kinds of systems, be it biosystems, RNA, proteins, and so on, but also for ligands to split those up in an automated way. You can, of course, go and do it in a way as you wish, but you can also do the automation. So we have examples here which also on that Jupyter notebook level already visualize these heat maps. So I encourage you to, if you're interested in this topic, please go to our facts homepage, to the Orca Python interface product page, and there you can directly get access also, or get directed to the corresponding GitHub page. So this was about ribonuclease. I want to show you one other protein ligand example, which is KRAS. So KRAS is not a problem, but it's an important target for drugs, because it is prevalent in cancer. So usually there is one amino acid in KRAS, which is a glycine residue, and when that mutates to cysteine or aspartate, then that's when cancer starts to develop. And so this is a popular target to really inhibit this type of protein with that mutation here. And so covalent inhibition is something that seems to be very promising for KRAS. And what happens in covalent inhibition is we have a ligand which binds not just with non-covalent interactions, but which binds covalently to this target protein, and that way inhibits it so that it can't make the bad action that it would otherwise make. And what we developed, and I'll show a little bit on that later also, is we have a conformational search tool within ORCA. We have these multi-scale tools, and we can now also run conformational searches in an entire protein. So we can take a ligand and we can try to investigate which are the most stable conformers of a particular ligand in the protein. And this can all be done with ORCA, and also we have a Wiesel tool, a workflow solution tool, which I will talk about in one of the next slides. And that can, like our conformer search tool GOAT, can be used to create possible conformers of the ligand in the active site. And then what we usually do in our standard procedures is we do optimizations on different levels of theory, and in order to get to a final realistic ensemble, which is something that really is reflecting what we have in the experiment. And so that way, for this case here, we end up with 3 conformers. This is a number, 3 conformers is much less than we usually would have for a drug in solution, because in a protein, we have like a lot of constraints from the environment. So it's of course also quite interesting to check, okay, how does the lowest energy conformer in the bound state compare to the lowest energy in the solution state, lowest conformer state in the solution energy. And that is giving you the distortion energy or the preparation energy to get your ligand or your drug from solution phase to the protein state. And covalent inhibition includes that we also are forming a covalent bond between the inhibitor and the protein. And that's something that you can also compute with ORCA. So with our QMMM tool, multi-scale tool, we can combine that with our so-called NEBTS method. NEBTS is a method that you provide a reactant, a product state, and it gives you the reaction path and the transition state, and that way also the activation energy for this reaction. And you can also run that in a protein. So that way you can analyze and predict how does this reaction take place, and what is the activation energy that I have to put into that system. And you can also, with these LED approaches, try to understand what is the effect of the enzyme, or what's the effect of the protein on this activation energy. So that's the first part of my story. The second part, it's a bit smaller, but it's something that I also brought and where I'm happy about from industry partners. So I want to show you some real life examples where these high accuracy methods that I just showed you and explained to you are having an impact in industrial R&D. And for this, maybe as a short preparation and teaser, we learned quickly when we founded FACCTs and started to work together with industry that ORCA is a very efficient tool and gives really good results. But we also learned quickly that industry people don't have much time. So they need standardization, they need workflows, and they need results. They don't want to fiddle around with these quantum chemical calculations. And that's where we started to develop our workflow solution tool, Wiesel. So what does Wiesel do? It has built in a lot of standard workflows for different use cases, like spectroscopy, reactions, process chemistry oriented, but also for drug discovery. And for these workflows, it has robust defaults, and it can be used easily. So it takes away all the complications, like that you need to choose the right method, and so on. So we provide best practices within these workflows. And another nice aspect of Wiesel is that it also does the quality management. So if something goes wrong with the quantum calculation, that Wiesel is taking care of that. And also what it does, it fully automates these entire workflows, and you can be used in a local cluster, but also in the cloud. And last but not least, if you have a high throughput run on a lot of different systems, it's quite useful if you have standardized output. And so that's also what Wiesel does. It organizes the most important results so that you can easily compare. And that's something that our industry partners are using. That's just some feedback from our industry partners. And here's just one high-level workflow example. So what we do is we just put in a structure file that can be an XYZ file, a small string that can be an SD file, PDB file, and so on. And then, for example, Weasel does a conformational search, creates a realistic conformal ensemble, and based on that conformal ensemble, it produces a realistic spectrum, for example, for NMR or for ECD and other areas. And now that's something that people use, and that's how people in industry use, and that's how they can combine these data also together with the experiment, or try to do a pre-screening, a pre-selection of compounds, first run the computations, and then just take the most promising candidates and then do the actual experiments on these selections of high promise candidates. And here you see on the upper side here, you see a depiction of like all different phases of ligand development. And I want to show you on the next slides that these tools are used in all these phases of ligand development. And so phase 0 is like the very early part. This is for not for a drug, but for an agrochemical compound. So pesticide. And here for pesticides, photostability is important. Like pesticides, like other agrochemical compounds, they are put on the fields, and these fields, they are like wherever agriculture is. And so everybody's also in South Spain and in Brazil, where there's a lot of sun burning off the field. And so that means these compounds need to be stable. You don't want them to degrade so that they don't lose their efficiency and that they also don't create a harmful product. And so at an early stage, you want to really understand how photostable is my compound. And what we did here together with our partners at Bayer, who provided these data here, we developed protocols which try to predict the photostability of these compounds. There's different possible mechanisms, like really chemical and electronic mechanisms, how these compounds degrade. And here we just show like a way of like on a very high level that we can take excited state results. So excited states is another method in quantum chemistry. You cannot just compute like how does a molecule look like in solution, but also when you add light to it, when electrons come in and excite, or when photons come in and excite electrons to an excited state, which is also something that we see in OLEDs or in LEDs when you're watching TV, when you're looking at your phone. Also in that case, excited states are important. So here in this case, we looked at different properties like excited state, energy, spin orbit coupling, and so on. And we correlated those with experimental data from photolysis experiments. And these photolysis experiments, that's the half-life of the photolysis experiments, they give us an indication how long such a compound would survive on the field. And on that way, you can also take these descriptors that we compute and combine it with machine learning together with the experimental data. It helps you to predict how photostable compound is. And here, in our company name, it's fast and accurate. So we want to get an accurate result that's most important, but also at a high speed. So we really tested a lot of different levels of theory. So we talked about the semi-empirical data up to couple cluster, which span orders of magnitude of compute time. And we found that here really an accurate QM level is important for these excited state energies, because excited state calculations, they are kind of complicated and electrons are interacting partially in a weird way. And we found that we really need some accurate QM level like hybrid DFT in order to get to meaningful results that are then also useful so that you can predict something, because only if your result is accurate, you can make use of it. Otherwise, you just don't need to run that calculation. So that's an early stage area where high accuracy calculations are of importance. Now, this was one aspect. What you saw was one aspect how we can predict photostability. There's other aspects or other mechanisms how photo degradation can take place. And one other aspect is that sunlight can excite oxygen to become singlet oxygen, which is highly reactive. And what we have within ORCA available is our conformal search tool that's called GOAT. And this conformal search tool can also be used as a reaction exploration tool. And so what we did in this workflow here is we take singlet oxygen and we let it react with, for example, with the diazepam here. And what goat react gives you is it gives you all possible reaction products. And then what we can do next is we have these reaction products and we can compute the thermodynamics, but also the activation areas when going from reactant to product. And this gives us an indication which of these reactions are actually accessible. And that means we need to have a thermodynamically favorable reaction. So the reaction energy must be negative. And also the activation barrier, that's what you see here, can't be too high. And so that's another area where this can be used. Now, going to a bit later stages of development, it becomes also important now when I swallow a drug or when there's some traces of agrochemical compounds which also get digested by humans. What happens to these compounds in the human body? So they are usually metabolized. And so we need to know what are possible metabolization products of these compounds. And so here, for example, an established way is to use Fukui functions. So Fukui functions is when you add you take your molecule and you add an electron, or you remove an electron, and you check, okay, how does the electronic structure of this compound change when doing this. And this correlates usually quite well with possible metabolites. And that's another area where high accuracy QM is used. Now let's go, in the interest of time, to the next example. So this is at phase 2, phase 3 of development, when really we go into the synthesis of compounds and maybe also intermediates of a synthesis and when there's also regulatory needs to understand how does a structure look like. And so there is, not sure whether everybody here is familiar with stereochemistry, but molecules can have stereocenters. So there can be, depending on how the bonding situation in the molecule is, and the authorities, they need to know about the stereochemistry of the molecule. And that's also called absolute configuration. So that's something we see on the next slide. And usually you get information about this absolute configuration of a compound by X-ray crystallography. But sometimes you can't crystallize such a compound. And so if there's no crystal structure available, you can also deduce the experimental structure. So that's not the right path to get this information. We know, or a lot of people here probably know, that ECD spectroscopy and VCD spectroscopy, they can distinguish between different enantiomers for such a chiral compound. But these spectroscopies, they don't tell you which actually is the current enantiomer, so whether it's an R or an S enantiomer. And that's where really the advantage of these simulations come into play. And so what we developed over the last years is protocols which make use of the conformational search that we had a look at. And then based on this realistic conformal ensemble, also we run spectroscopic calculations, for example, ECD calculations or VCD calculations. And then we compute so from the complete conformal ensemble with these spectroscopic properties for each conformal, we know about the conformational energies, so we can compute a Boltzmann-weighted spectrum, which then we can compare to the experimental spectrum. And that's something that you see here. So here for an S enantiomer, you see the simulation here in red and the experimental result in green. And here you see the corresponding results, experiment versus simulated, also for the R enantiomer. And so that's something that people are now using in a standard way in order to identify the absolute configuration of the compound in case crystallization is not possible. And here I want to emphasize, to get to the right result, we need complete accurate conformal ensemble, we need the right energies for the conformals, and also good spectra. Now, if we go, now we stay in this stage here, then also when we go from like really small volume to larger volume, where we need more and more of the ligands that are produced in order to run the experiments that are necessary to go on to the next steps. Often in the lab, the synthesis routes need to be optimized. And And so here and so it also really becomes important to make these processes more efficient. And so here is an example that's about the cycloaddition of this compound, which the cycloaddition also produces a carbon compound. So that can be an R or an S enantiomer here. And so now in this example case here, there was the R compound was about 25%, and the S product was 75% from the experiment, and that was unfortunately like the wrong ratio. So what they needed was the R compound, the R enantiomer. And so what they did also now on the computer, they computed the reaction mechanisms, and also with checking both different routes and having accurate energies, accurate activation energies, you can also compute these ratios. And the model was right because like the model was good, so they could reproduce the experimental result, and then they could play around on the computer with the models with different additional reagents in different experimental conditions, and found based on that understanding, found a way to invert that ratio. So having almost 100% R enantiomer and just 5% of the S enantiomer. And that brought these suggestions then to the lab, which could also be reproduced in the lab, and which helped then the overall process to get to the right compound in the most efficient way. So now, and here also I want to emphasize, you see here these delta G values, like to change this ratio is really a matter of kilojoule per mole. And that means we really need a sub-kilojoule per mole accuracy in order to have a high predictive capabilities here. Now, this is my last example. This is from very late stages of development. This is actually when pharma talks to the authorities, there's often like a lot of data that's required. This is a case where that was a market product also from Bayer, and the authorities wanted to know about the binding mechanism of a market product, how it binds actually to P450 enzymes, and for the compound itself, there were no X-ray structures available, which was a problem because the authorities wanted to have these data. And so what happened next was they were running couple cluster calculations on models of this protein together with the ligand in different possible binding modes. And so that's what you can see here. You can check how does my ligand bind to the heme center here, for example, with the nitrogen or with the sulfur. Then you can compare these binding energies. And given that couple cluster calculations are established to yield high accuracy, these results were also then accepted by the authorities and helped in the re-registration process. So I want to summarize here. I hope I could show you that high accuracy quantum chemistry is nowadays possible for real-life systems and life sciences. The energy decomposition scheme that we develop provides really valuable insight into molecular recognition. These high accuracy calculations can have a big impact on all phases of ligand development, and they can help also our pharma and partners to provide crucial data to the authorities and also to secure the IP. I want to quickly thank the people who were part of this, so especially Bayer, Andy Beck to provide some of the slides and use cases, and also our collaborators within Perugia who also helped a lot in the covalent development, the FACCTs team, and our collaborators within the Max Planck Society. And I want to thank you for listening and wish you also fun. If you're